So let's move on to the next speaker. Next keynote talk will be given by Dr. Professor Guojun Sheng. Dr. Sheng obtained his PhD in developmental genetics from the Rockefeller University in 1997. He directed the laboratory of early embryogenesis at RIKEN, Center for Developmental Biology in Kobe, Japan, from 2004 to 2015. Now, Dr. Sheng is a professor at the International Research Center for Medical Sciences and the Graduate School of Medical Sciences in Kumamoto University in Japan. His laboratory uses the avian animal model and studies cellular and molecular mechanisms regulating cell fate changes during early embryonic development. Today, the title of his talk is Omics, Resources and Tools in Underrepresented Animal Models, a bird's eye view. Dr. Sheng, please.
Thank you very much, Professor Nakai. So I will start screen share. Let's see. Can you see?
Yes, we can see.
Great, fantastic. Okay, so thank you very much. And we do a lot of bioinformatics and genomics, omics related work, but normally I do not attend bioinformatics meetings because our primary interest is not on the tool itself, but rather to utilize the tool for our developmental biologist study. So I consider myself as a developmental biology with an interest on a few other aspects of things related to developmental biology, including cell biology, evolution, and of course the general understanding of comparative anatomy and association of the developmental biology to disease. So I try to use this little guy as an example. So we all know birds are actually dinosaurs, right? Relics of dinosaurs. So dinosaurs are not far away from actually humans. So I will try to tell three little stories about what we do, and one additional one with a little bit of relationship with the omics related work. And then in the end, I try to convince you that actually we need expertise in bioinformaticians to actually solve some key issues that of interest both to us and to people who might be more interested in cancer research. So we know that chickens are actually reptiles, and together with mammals, humans are mammals, they constitute a little branch called amniotes. So amniote is one branch of the so-called vertebrate tree here. And if you were to do a little bit of a kind of a molecular phylogeny, the amniotes evolved around 330 million years ago, and they can be separated in two major branches. One is the so-called reptiles, the other is the mammals. In the mammals, we belong to this little part is called eutherian mammals. And the birds that we use as a model system belong to this part of the reptilian tree here. So this little branch of the animals, they're quite different from others in the sense that they develop on land. Actually, they are not water-based. So because of this mode of development, there are a lot of changes and modifications in embryogenesis which have to accommodate this drastic physiological change during early animal development. So accompanying this type of evolutionary change, they have the similar genetic makeup because they have similar kind of origin, evolutionary. So birds and mammals have very similar genetic makeup. And with the similar genetic makeup comes, of course, the molecular conservation, signaling conservation, and cell biological and other aspects conservation here. So we are a part of animal called chordates, and they are actually from molecular phylogeny analysis, there are so-called 17 ancestral linkage groups of all chordate species in a way. And at the early evolution of the vertebrate, there were 2 rounds of whole genome duplication. So from 17, involving 1 fusion event, so there are about 54 chromosomes, ancestral chromosomes in vertebrate ancestors in a way. And then they evolve a little bit in the amnio branch. There are actually 49 ancestral chromosomes. and due to additional shuffling, including fusion and fission of this certain chromosomes, the actual chromosome number in human and in chicken, of course, are quite different, but they can all easily be traced back to ancestral amnio chromosomes, 49 or so. And changing chromosomal number doesn't change much in terms of their actual gene content. They could be located in two separate chromosomes, but their genes, intron-exon boundaries, etc., are quite conserved in a sense. So the chicken genome was sequenced in 2004 in comparison with the human, which was sequenced 2001 here. And chicken has 38 autosomes and one pair of the sex chromosomes here. In comparison, chicken has 22 haploid chromosomes and X or Y. Birds, they use actually so-called ZZ is the actually male, ZW is female, so it's somewhat different from mammals in a sense. And chicken genome has a 1.1 billion base pairs, a little bit, a third of the human gene content. But actually number of genes are very similar, about 20,000 or so, and number of non-coding genes are also very similar in a sense. And the current assembly is Gal-6, and human has 36. So this kind of somewhat reflects the effort of the community in the sense there are very few and far between updates in the genome assembly. And the result of this is that the sequence information of actually 6 chromosomes are still missing. And you may find it a little bit strange, but actually birds have a lot of so-called microchromosomes. They're very small, and it's actually very difficult to piece them together in terms of the sequence. So actually 6 chicken chromosomes information in terms of how they are organized in the genes, gene contents of these chromosomes are still missing. And also additional data on cell and tissue specific transcriptomes, transcription initiation site, promoter, enhancer information, splicing, non-coding RNA, DNA methylation, genome annotation, etc. Are very primitive in comparison with what we know of the human genome itself. But this probably is not unique to chicken. In general, it's unique to animals that people use as a model system, but they are not a major type of animal models in a way. The chicken is pretty well used for developmental biology, but because of the community size, actually, the effort to improve those omics level information is pretty primitive here. And this, of course, is reflected by the fact that the community size and the funding opportunities are very limited in comparison with the available humans. But we still need to, work on this. So why do we study birds, right? So there must be some advantages of using the bird species as an animal model. And just to give you a broad overview, is that actually avian species contribute to vertebrate biodiversity in a pretty significant way. So currently it's over 10,000 living avian species, right? And although 4.5% of those are listed as an endangered species here. And in comparison, all the mammalian species combined, there are only about 6,400 or so. And the majority of those are actually belonging to groups of rodents and bats, very speciose groups. The rest is actually not very biodiverse in a sense. So, but birds is a very successful group of animals and occupies many different eco niche in a way, so that you have 10,000 more living species here. And they contribute to human health and human well-being in a pretty significant way. And everybody may be eating chicken meat and chicken egg every day, actually. So if you look at it, we have over 70 billion chickens being slaughtered for the purpose of chicken meat in the world annually, and over 80 billion kilograms of the chicken eggs being consumed in the world. And these are, of course, contribute significantly to the nutrition and of daily life of human society. And from a medical point of view, health point of view, every year people will get flu shots, and most of the flu shots are actually produced in chicken eggs. You probably don't know that, but it's still the best way to make the influenza vaccine and some other vaccines which are very important for agricultural purpose in a way. And in terms of science, it's actually, people don't know, but actually the oncogene, the concept of oncogene was developed in the chicken. And the gene itself was discovered in using the chicken gene as well. So over half a century ago, the Peyton Rouse discovered the Rouse sarcoma virus, and it's an avian virus. And the gene that is responsible for this viral transformation of the cell was actually also discovered in the chicken context in a way. So we are developmental biologists, so we are interested in this model primarily because of its value for developmental biology. So I will give you a few examples to see what we do using this model. And in the end, I'll try to convince that actually developmental biology and cancer biology and in bioinformatics are actually could be smoothly merged in the sense to solve biological questions, problems that will be of interest to the society in general. So I'll show you two movies here. So these are the avian embryos. So this is actually, on the left side is quail embryo, on the right side is a chicken embryo. So give you a sense of the dynamics of the development processes that we're interested in. So within 2 days period of time, you can see the chicken egg, the quail egg itself here undergoes pretty dramatic dynamic change. By the end of the second day of development, you see most of the rudimentary tissues and organs are already present, and circulation, heart is beating, circulation is starting. So this is only about 2 days of animals develop in a sense, from more or less nothing. No structural, recognizable structural element to a very well recognizable structural anatomical element here. In the next 3 days, from day 2 to day 5, you will see a more dramatic change in a lot of things, including the maturation of the brain tissue, the vascular systems, and the limbs, the forelimb, hind limb, and the tail bud, for example, and eyes, and all these sensory organs are developing in the next 3 days. And remember, this is only first 5 days of the about 3 weeks of the chicken development. So these are the things that we are interested in, we study. And there are a special group of scientists they call developmental biologists. They are interested in various aspects of the animal development, whether they use chicken or mouse model or some other animal models. There are some conserved mechanism that we can understand that's applicable to multiple different species, as well as to the homeostasis of the mature tissues and organs in human in a way. So our lab uses avian model to study a particular cell biological process called epithelial mesenchymal transition. So the reason we are interested in is that we are multicellular system. Our body is multicellular, and in the multicellular system, the cells have to be organized in some sort of organization. Actually, there are only two types of organization. One is epithelial, one is mesenchymal. If you think about that, most of your inside your body are compartmentalized, and the compartmentalization takes the epithelial structure here. So, so you have the transition in development as well as in the homeostasis period of the adult life, from an epithelial to mesenchymal, or the other way around, from mesenchymal to epithelial cells. Of course, these are cell biology, and there are some complicated cell-cell junctions, cell subcellular organizations that are required to maintain either the epithelial feature of the multicellular organization or mesenchymal feature of the multicellular organization. So I will just very briefly go through 3 aspects of the EMT that we are studying using the chicken model, and then I will show you one examples of how we incorporate the omics informatics in our research, and then I will finish with a call for participation of all you, mainly bioinformaticians, in actually cancer EMT research. So we are studying developmental EMT research, but cancer EMT originates from the tissue, which actually, they are derived from a developmental, progress in a way. So they still maintain certain cell biological organization, which can be understood by studying that development of those tissues and organs. And some aspects of that really takes the participation from people like you in order to get a better understanding of what we can extract from the information of the cell biology here. So early development of births, first example is so-called gastrulation EMT. So this is the chicken egg. And the embryo itself is the blue dot here. And if color is artificial, of course. If you zoom up and you see a structure like this, so by this time, there's nothing you can see, but of course there are many cells already. And if you do a cross section of this structure, you will see actually just a thin layer of epiblast here, so-called. And these are pluripotent, meaning that they are, can give rise to multiple, almost all the cell lineages in your body. And then in the next half a day or so, these cells will undergo morphogenesis, create a structure called a primitive streak. And the primitive streak will actually be the center of the so-called internalization of the cells that are coming out from the outside and going into the inside of the embryo here. So remember, we have a body is a multicellular system. It's very complex in its organization. And the initial phase of the generating this complexity is by moving cells located outside into the internal space of the embryo. And this famous scientist called Louis Wolpert once said, it's not your birth or marriage or death, but your gastrulation, which is truly the most important time in your life, because that's the time when you generate so-called 3 germ layers. And from each germ layer, all the tissues and organs of your body will be derived from. So the actual process of gastrulation, I will show you in a movie here. So this movie is about only a short period of time, 6 hours or so, but the cell biology of the gastrulation is very dynamic. So you see, if some of these cells are labeled, you can see that in a few hours' time, these cells will move from the epiblast, peripheral part, into the center part of the epiblast, and then they undergo epithelial to mesenchymal transition, and then they migrate out as individual mesenchymal cells here. Now, if you do a cross-section here, you can see the schematic representation like this, is you have the epiblast cells, and they undergo epithelial to mesenchymal transition. They can become mesenchymal cells, and they migrate away. They'll contribute to different parts of your body, mesoderm lineage of your body here. So it's an epithelial to mesenchymal transition. We can study it by looking at the gene expression and by looking at their cell biology of that, polarity of these cells. So because we are studying epithelial to mesenchymal transition, we spend a significant amount of our lab's effort in trying to understand how the cell and intracellular organization of this epiblast cell and interaction of the cell with the extracellular matrix basement membrane here is affecting this process of the epithelial to mesenchymal transition. We've been focusing on the function of the basement membrane and the integrity of the basement membrane, as well as interaction between the basement membrane and the cell, basal side of the cell, and this intracellular cytoskeletal elements here, microtubule in this particular case here. So we came up with a model is that the maintenance of the microtubule integrity is essential in the maintenance of the basal cell membrane and the basement membrane, which is extracellular structure here, and involving basal cortex and transmembrane proteins like dystroglycan and integrins in here. So this is the one aspect of the thing that we study. And the second aspect of the EMT we study is so-called pluripotency, something to do because the gastrulation is a process where a pluripotent cell population will give rise to lineage restricted cell population here. And the first phase of lineage restriction is generating from the epiblast to the ectoderm, mesoderm, and endoderm, 3 different germ layers here. So this is a brief summary of the actually chicken early development from a few cells. So chicken is like all of us, starts from 1 cell and undergo division into a few cells and into many hundreds of cells, and in the end have many thousand cells. If you do a EM electron microscopy section, you will see that the chicken yolk, egg yolk is here, and the cellular components of the chicken embryo is like this. You have the contact site between the embryo and the yolk, and you have the part of the embryo that will eventually give rise to all cell lineage in your body here. And this kind of organization is actually similar to what we understand as mammalian ones. So this is an example of the platypus. Platypus is a primitive mammal, and they have pretty much the same kind of organization as the chicken embryo here. So the mammals are derived from type of organization that we understand in the chicken embryo here. And this is a comparative comparison between a mouse or human embryo versus birds and monotreme embryos here. And they can be traced to a similar type of a general multicellular organization, which I will not go into detail to. But the point is that we want to understand whether the pluripotency of the epiblast has anything to do with the so-called epithelial or mesenchymal feature of this epiblast here. And our theory has been that before the before the gastrulation, before the 3-germ layer formation, the epiblast has to be epithelialized from a partial epithelial to a full epithelial structure with the apical, lateral, and basal membrane compartmentalization. And this epithelialization is essential to leave the pluripotent state of the epiblast and to initiate the so-called 3-germ layer formation here into the ectoderm, mesoderm, and endoderm from the pluripotent epiblast phase here. And we can draw a comparison between what's happening in the chicken embryo versus what's happening in the human embryo, and they more or less undergo the similar kind of transition from a naive phase to a primed phase and to an exit of the pluripotency phase, which is correlated with the epithelial status of these epiblast cells here. And you can schematize it with this sort of landscape here. You have a naive phase, formative phase, primed phase, and exit phase. And then you will undergo the differentiation of ectoderm, mesoderm, and endoderm. So the epithelialization process is actually quite critical in transiting from primed to exit phase of the development here. So I will very quickly skip to the third one, which is something to do with the placentation, actually. So as I said, birds and mammals are derived from the same kind of organization. And although we develop in maternal environment, while eggs develop not in the maternal environment, but the embryonic architecture is very similar. And the placenta we know of in the mammals are derived from a structure called CAM. CAM stands for chorioallantoic membrane. And that's the fusion of the chorion, which is this part, number 2 here, and allantois, which is number 4 part here. So these two tissue architecture will fuse to form a fused so-called chorioallantoic membrane. And this is the precursor for the mammalian placenta here. So the function of this is to mediate, in the bird's case, to mediate oxygen uptake and calcium uptake from the eggshell, oxygen from the environment. And the same kind of function for human placenta is to mediate nutritional uptake from the maternal blood, as well as the waste disposal from the embryonic development here. So the origin is very similar, and we are interested in the epithelial to mesenchymal process of these two tissues here. I will just show you the movie again here. So this is the chicken embryo, and the allantois comes out quite late. So at this time, allantois starts to come out as a little balloon here, and the balloon will actually rapidly expand until it reaches the chorion here. And when it reaches chorion, it starts to fuse with the overlying chorion. And this interface after fusion will be the interface for the oxygen uptake and calcium uptake for the development of the chicken embryo here. And the very similar function, as I said, for the placenta in mammals in a sense. So we were studying how the epithelial to mesenchymal transition of these two cell lineages mediate the fusion of the chorion and allantois here. In terms of how it regulates apical basal polarity of these two mesothelial layers here, right? And this placenta, of course, placentation is similar, involves similar kind of cell lineage here. So this is in case of a mouse embryo here, you have the allantois coming out and merging with the chorion, forming a placenta here. And in the human case, it's more or less the same kind of mechanism in a way, right? So going back to the genomics aspect or omics part of that. As I mentioned, the information in the chicken in terms of the tissue and cell specific transcriptomes, initiation site, etc., is very scanty. So we actually utilize a lot, and in this one particular case, we try to map the transcription initiation site using this in collaboration with the Recon Omics Center. Map the transcription start site or all the genes of the chicken embryos, which a paper published a couple of years ago, is a systematic analysis of transcription start site in avian development. This allows us actually using the technique so-called cage. So I will not go into the details of the cage technique here, but the idea is that the information here is actually very useful and allow us to manipulate the gene expression in a way that we cannot do, because remember the chicken embryos, we cannot quite do genetics because it takes too long time to do genetics with the chicken embryo in a way. So we can utilize the information of the transcription initiation. For example, this is of course, a very housekeeping gene, JPDH. And there's a little peak here, and the unique peak here at the very beginning here, which means that this particular nucleotide is the initiation site of this complex many exon gene in a way. And we can look at this type of aspect in many different genes, and we can understand their expression variation, as well as to precisely localize the transcription start site, because the ESD-based mapping often does not give you a very precise 5' end of those genes here. Without precise knowledge of the 5' end, oftentimes lots of the gene annotation itself may be wrong, because they could be missing a particular exon which is located in the 5' end. And if you were trying to make a cDNA out of that, it's probably giving starting off with the wrong information here. But with the TSS knowledge, we can actually very precisely know that what's the initiation site of a particular gene. And we can also quantify the housekeeping genes, what exactly can be utilized as a housekeeping gene. For example, GAPDH is here, and beta actin is here, for example, here. And even for housekeeping genes, we know that actually is a huge variation in terms of the gene expression level. So this is kind of cautionary tale for what people use as a housekeeping genes here. Or the alternative promoter usage. In this particular case, a gene called CDYL has two promoters being used, TSS1 and TSS2. TSS1 is somewhat ubiquitous, and TSS2 is very tissue specific. So this happens with a lot of genes, that you have multiple promoter usage, as well as the tissue specific usage of promoters here. And with this knowledge, we can actually try to activate and inhibit the gene expression using this technique called a CRISPR-mediated gene activation, a CRISPR-mediated gene inhibition here, transcription inhibition here. So I will not go into details, but knowledge is that the idea is that you need a very precise understanding of a transcription starter site. And then you can use this knowledge to actually target using a guide RNA in this area, and to activate a gene or inhibit the gene. This is actually generalizable for any kind of species. I have the chicken, so anything that cannot be easily utilized with the genetic or molecular studies, you can use this technique to actually turn on or turn off a gene here. So the last 2 minutes or so, I'll just generalize with cancer, right? So we study EMT in the context of developmental biology, but cancer people are very interested in EMT. And the reason is that the majority is that cancer is essentially a problem with epithelial maintenance in your body. Actually, almost 95% of cancer in your body is actually originated from epithelial structure here. And a minor case like blood cancer, for example, is not epithelial structures, but majority of the cancer originate from normal homeostatically maintained epithelial structure. If something goes wrong with the maintenance of the epithelial structures, and you have the problem of EMT, you have problem of metastasis, you have problem MET in the metastatic site in a way. So the cancer is essentially an EMT issue, and people in cancer biologists are actually getting more and more interested in trying to use developmental biology models to understand what's going on with this EMT, and whether we can actually quantify it with some sort of molecular or quantitative information to see whether you can see which cancer is bad, what kind of signature in the cancer that you can use in terms of the EMT related genes that might be giving some prediction of the metastatic ability, malignancy of these cancer primary tumors in a way. So in the past 2 or 3 years, there are quite a few publications, this one included, to associate the metastatic ability with the EMT status of cancer cells. So they can identify a transitional states of tumor using a special EMT type of markers to see that if a cancer cell has these type of EMT marker, they are more malignant than other type of cancer cells, even they reside in the same primary cancer position, primary tumor location here. So as we said, the development of EMT has also this kind of issue, and we can, for example, the first case I explained is that you have the gastrulation EMT here. This is nicely epithelial cells. This is nicely mesenchymal cells, but in transition, in the middle part, you have partial epithelial cells and partial mesenchymal cell status. And oftentimes, both in Prepotency regulation, in stem cell regulation, as well in cancer metastasis cases, actually those cells are the most worrisome cells in a way, rather than the perfectly epithelial or perfectly mesenchymal cells. If you have a cancer cell, which is very, very mesenchymal, moves a lot, it's actually not bad. But if you have a cancer cell, which is partially epithelial or partially mesenchymal, those are the bad tumor cells in a way. And they most likely they are easily negotiating with the environment in the case that they are able to intravasate or extravasate and move to a distal place in a way. So there's some special features about those partial EMT, which can be, which people still don't have a good understanding of. Of course, you can hypothetically say that you have a perfect epithelial and perfect mesenchymal, and then you have partial status or partially epithelial and partially mesenchymal. But how do you well define them? What kind of score you give them? What kind of molecular signature you can define to say that this is partial status 1, partial status 2, partial mesenchymal status 1, partial mesenchymal status 2. And if you have a heterogeneous tumor cell compartment, if you do a single cell level omics, will you be able to use a bioinformatics information to say that whether this primary tumor is benign or whether this primary tumor could be developed into a malignant tumor very soon in a way. So we need more bioinformaticians to better characterize cells that reside in the so-called transitional states of epithelial to mesenchymal transition, both for the understanding of developmental EMT as well as for the understanding of cancer EMT. So I think if you are interested in any of these things, you can talk to colleagues. I'm sure in most universities, you will have colleagues working on the epithelial to mesenchymal transition, whether from a developmental perspective or from a cancer biology perspective here. And you can learn more and see whether you can participate in this kind of research. So I'll finish off by acknowledging colleagues and funding sources which have supported our work, and I will take questions if there's any question from audience. Thank you very much.
Thank you very much, Dr. Sheng, for very excellent talk. And so the questions or comments, please. One or two already appears in the chat. Dr. Sheng, could you see the chat box?
I'm trying to, do I have to stop sharing or?
No, that's not necessary. In the bottom of the Zoom, there is a chat.
Okay, let's see. Which is the first question?
Probably the
how much is it about Z and W? Is it first question?
Yeah, I think so.
Okay. So how much is the Z and W different from XY? So Z and W, so you can actually kind of trace back to the original chromosomes, and ZW and XY are originate from different chromosomes here. So, but actually this, the sex determination is actually a totally different research area, and then actually you have sex determination that is not genetic based. So you don't really need to have a genetic difference to create either a male or female. But in the case of birds, in the case of mammals, it's genetically specified. You have a sex chromosome, genes that might be regulating the identity of the gender for a cell or for the gonad development here. So the answer is that yes, this XY chromosome and ZW chromosome originally from different ancestral chromosomes in a way. Why we highly encourage?
This is not that question, I think.
Yeah.
Before seeing the next question, let me ask one naive question. Maybe I misunderstood your explanation, but according to your explanation, the EMT is related to cancer formation. So I wonder if mesenchymal cells are more similar to cancer cells than epithelial cells.
So that was the original idea, but now people, more and more people believe that these are not the perfect mesenchymal cells. So it has to be, so you would normally imagine the cancer starts off with some malfunction of the epithelial cells, and they have to move around in order to metastasize. So people initially think that the more mesenchymal cells are like bad character of the tumor. But now people realize it's actually not. So the more mesenchymal cells are actually not very deadly, but those cells which with a partial epithelial status, meaning that if you have a primary tumor, and if you can categorize the features of those primary tumor cells, those cells with the partial epithelial status seem to be the worst population of cells in the primary tumor. Of course, they could be genetically based or epigenetically based, meaning that this maybe interconvert between different epithelial and mesenchymal status, or they could have some sort of genetic element that some type of minor mutations in the primary tumor population will confer more mesenchymal status or more epithelial status to this heterogeneous tumor cell population. But in general, people believe maybe epigenetic regulation is somewhat more important in determining the metastatic capability of those primary tumor cells. Yeah.
So we are interested in epigenetic changes occurring on the genome. So I wonder what kind of changes appear at the time of EMT or cancer formation on epigenetically.
Oh, so anything that people study in the epigenetic regulation are involved. I mean, I'm using our own example. So, so we are looking into, in collaboration with the cancer biology lab, we're looking into the DNA methylation and how it actually help the EMT transcription factor snail in regulating its target gene. So the epigenetic regulator have an action on the so-called EMT regulator. So EMT regulators are a set of transcription factors, a limited small set of transcription factors which actually broadly regulate the cell biology of these epithelial mesenchymal cells. And these epigenetic regulators obviously have some role in controlling the regulatory behavior of those transcription factors. So yes, there are, if you look, there are many genes being affected by working on these so-called EMT transcription factors, right? So if any model you look into cancer biology-wise, you can actually put some correlation with the epigenetic regulator as well. The debate in the field is that which is playing a stronger role, whether it's purely epigenetic regulation or mutation-based, somatic mutation-based. Of course, somatic mutation is very important for the initiation of the tumor to start with. But in terms of the transition from a tumor, a primary tumor, a benign tumor into a metastatic tumor, we think EMT is a very important component. And what is the driver for this EMT transition is not very clear whether it's epigenetic or mutation-based change.
No, very interesting. Thanks.
Yeah.
So only thanks. No more questions. Any questions from the floor, audience?
Yeah. So maybe I will make a little kind of final encouragement slide. So one is that we are organizing a meeting on human development, which actually going to be held next year in Japan, in Awaji Island. And I think we will very much welcome participation of the bioinformaticians in the sense that in terms of the human development, which looking at the angle of the trying to recreate functional tissues and organs in vitro, using the developmental biology knowledge. And I think bioinformatics plays a very significant role in categorizing what kind of tissue and cells being generated in vitro. Similar to categorization or complexity of the tissue cells in the cancer case, in a sense. So, if anyone is interested in that, and also in general, if anyone is interested in collaborating with the cancer EMT or developmental EMT people, I'm sure you can know more a little bit by reading some of the papers, and then you can find a collaborator in your own universities or institutions.
Okay, thanks. If there are no more questions, so now
okay, I'll stop sharing. Let me see.
Thanks. Yeah,
Sorry,
we have another session for photo taking. So please, thank you very much, Dr. Sheng. Let's thank the speaker again. So we can't, you can't hear the applause, but we really thankful for your talk. Thank you very much. So I'd like to close the session.